My name is Diana Kuletsky. I'm working for Janssen's Immunology Translational Sciences, in particular there in the Digital Health Group, and I'm responsible for all co-diagnostic development activities within immunology. And I've been working for Janssen's for the last, I think, 15 years, having been in particular in diagnostic development, but also having experience in drug discovery research. So I know, so to speak, both worlds. And with that, let me start that presentation for that webinar. In the following, I would like to guide you through the journey of a gene expression signature we have developed at Janssen's Immunology in collaboration with one of our vendors. Over the years, many colleagues at Janssen's have actually worked on this project, and it is thanks to their work and the patients who gave us permission to use their samples for research that I can share some of our learnings in this seminar with you. I will start with the early discovery activities of the biomarker and will then share our cross-functional team's thinking, which led us from biomarker discovery to the development of a fit-for-purpose biomarker test. I will provide a brief recap at the end of both sections. We have to go back approximately 10 years in time when biomarker research activities started as part of a phase 2 clinical trial for the anti-TNF-alpha biological infliximab, or also known as Remicade. We were looking for a way to move towards precision medicine and immunology, perhaps similar as what was going on in oncology at that time, so that we could ultimately move to better treatment outcomes for patients with inflammatory diseases. What we wanted to find was a biomarker which could predict treatment response to anti-TNF biologicals, or specifically predict treatment response to Remicade in patients with ulcerative colitis. To do that, we collected colon biopsy tissue from patients at screening, and this was used to generate an RNA expression profile, first on a microarray with 109 assays. True clinical treatment response was determined by endoscopy and histologic assessment of colon biopsy at week 8 in the trial. This means the mathematical algorithms were trained using endoscopy mucosal healing data at week 8. Several iterations and trainings of the algorithms were performed using data from a total 3 phase 2 studies until we came up with a refined RNA expression signature of 13 genes. From the expression signals of these 13 genes, a weight score is calculated, which predicts, dependent if the calculated score is above or below a certain threshold, if a patient will respond to therapy as assessed at week 8. In the table, you can see the 13 different genes which together are part of the RNA expression signature. They represent, in a nutshell, biological processes associated with inflammatory response. And in general, higher signals suggest a more severe inflammatory condition. The graph shows exploratory gene expression data from one of the trials with admittedly only a limited sample number. And the data suggests that higher expression values at baseline are more likely to occur in predicted non-responders, here shown in blue. This is a complex table and shows an overview of the different studies which were used to identify and refine the 13 gene expression signature and retrospectively assess our biomarker hypotheses. Before we go through some of the data, I would like to clarify that the assay conditions were not locked and that data had been generated using different qPCR technology platforms. As it turned out during the first studies, the gene expression signature, which was initially evaluated to predict treatment response to Remicade or other TNF-alpha biologicals, turned out to show a much better performance to predict that a patient would not respond to therapy. This is reflected in the consistently high negative predictive values across different studies. What does negative predictive value mean translated to our signature? It means that the negative predictive value is the percentage of patients with a negative test who do not have the disease, or in our case, the percentage of patients with predicted non-response who do not truly respond to treatment by means of endoscopic healing. This strong NPV is also the reason why, going forward, we use the term NRS, or non-responder signature. And as you can see from the clinical specificity in the various cohorts, the NRS identifies only a subfraction of the true clinical non-responders. What is, however, remarkable and came as a surprise is that the signature not only works for different TNF-alpha inhibitors, but across at least 2 additional mode of action biologicals. In the latest and largest study cohort, Unify here, we were able to include also samples from the placebo arm. And based on all the data we have obtained so far, it suggests that the NRS is a prognostic signature. Let me quickly recap. We have developed a 13-gene RNA expression signature, which performs well in predicting non-response to treatment. The gold standard or true test, which was used to train and develop the RNS for response prediction at baseline, is endoscopic mucosal healing or clinical remission at week 8. It identifies a subfraction of true clinical non-responders. And the data suggest that the NRS is prognostic. And from the data obtained so far, in particular from Unify, as I have just shown you, we have seen a higher prevalence of the NRS, or predicted non-responders, in bio-IR patients. The question was then, is there a value or utility for this biomarker for drug development, in particular, if we think about difficult to treat patient populations, and if there is a way to overcome current treatment obstacles with novel mode of action drugs or combinations thereof? Which brings me to the question, what were our thoughts and considerations regarding the NRS and how to use it, if at all, in other UC clinical trials? Well, dependent on the audience you ask and how to best proceed with a biomarker signature, you might very likely get the answer from one fraction that it would be best to continue retrospective exploratory research and assess if the signature works in other studies, and to further refine and improve the test by perhaps adding genes or changing the score, as there's always room for improvement. On the other hand, there was also acknowledgement from others that we had tested the NRS already retrospectively across several study cohorts, and that the value to continue doing so was seen as limited, in particular if we can't provide actionable information. So, in a hard-won consensus, we concluded that the time had finally come to boldly move forward and think about how could we utilize the NRS prospectively. What's the value of a prognostic biomarker? And which arguments could you use to weigh the risk or benefit of implementing a prognostic marker into clinical trials? The probably most straightforward and least risky approach, thinking about overall clinical operational impact, might be to go for randomization or stratified randomization. If the NRS would be used for randomization, we could mitigate the risk of an imbalance of patients who would or would not respond to therapy and enrolling them into certain trial arms. Another approach we thought about was enrichment. This means to have study arms enriched with predicted responders or non-responders, and which would help us to address patient segmentation for a certain mode of action drugs, and perhaps assessing a difficult to treat patient population, probably the predicted non-responders, who might benefit from combination therapy. But the question is then, what type of test would we need to develop to use it for such different applications? To visualize our thinking and, let's be honest, to enhance internal education and raising awareness for biomarker development aspects, together with colleagues from regulatory affairs, we came up with this decision tree. We first asked if the assay, or specifically if the test results, would be used retrospectively or for exploratory research purposes, or alternatively, if they would be used prospectively. If used for exploratory research purposes, a research use only test would suffice, and we could probably just keep using the research assay as it existed. If, however, a prospective use of test results would be foreseen, then the development of a clinical trial assay was required. This implies that for each specific clinical trial, a multi-parameter risk assessment needs to be performed, for instance, to determine if an IDE submission would be required. There are multiple other considerations to be taken into account, and I will come to them on the next slide. But part of the biomarker development strategy is also to determine if the data suggests that the biomarker test results are required for safe and efficacious use of the drug, and as such required for drug prescription, which would lead us into the arena of companion diagnostics. Or alternatively, if the biomarker test is required to be submitted for approval and being launched together with the drug, or if there are potentially other medical reasons which impact market access considerations to make a test available after the launch of the drug. In any of these cases, when the test needs to be available beyond clinical trials for the purposes I just mentioned, the development of an in vitro diagnostic test needs to be foreseen. And just to finish this thought, if an in vitro diagnostic test would be required, we would also need to determine rather early in the biomarker assay development trajectory how the commercial testing strategy would look like. Based on this example here from the US, the question would then arise, should one go for a single-site laboratory testing strategy with, for instance, a PMA in the case of a companion diagnostic, or would one rather go for the development of a commercially distributable IBD kit? Of course, in most cases, bridging strategies between the different tests, between the clinical trial assay used in prospective studies, the IBD test, or the IBD kit would also have to be foreseen. Given the complexity of biomarker assay and ultimately diagnostic assay development, and the associated financial risk to invest early in such a test development, it is probably fair to proceed with a cautious development and risk mitigation approach, which led us to consider a staged and what I would call fit-for-purpose biomarker development strategy. And if I may just go back for a moment, based on previous considerations, we had taken the decision to keep the research use only test to support our biomarker exploratory research needs. But in addition, we also decided to further develop the NRS as a clinical trial assay to be available for prospective use and to support stratified randomization and enrichment. However, to ensure as much consistency and comparability of the approaches, we do keep all NRS-related development activities under one large umbrella. As briefly explained before, we really had the idea to develop the NRS to a point of what I would call a generic CTA. This implies, of course, keeping in mind study-specific risk assessment, and that the test would need to be far enough developed to be used prospectively in different clinical trials with different drugs or combinations thereof. And one of the very first questions to ask is about the intended use of test results and in which patient population this may be used. We had previously observed that the NRS data from a study cohort of Japanese patients had behaved slightly different than other patient cohorts. This means that for the development, disease severity and genetic diversity of patients need to be considered. Beyond assay performance characteristics requirements, the control strategy is another important topic. In particular, as we know that we use colon biopsy tissue as sample type, and that previously screened cell lines had not been given sufficient evidence to be used as controls for precision studies. And if a test in ulcerative colitis is being used as one of the randomization parameters, turnaround time of the test becomes important, as does the envisaged sample throughput. To use a test for randomization and taking all clinical trial logistics into consideration, we know we need a turnaround time from sample to reported test result of approximately 3 to maximal 5 business days. This changes the game entirely and led us from population-based data analysis and batch reporting, what is fine in retrospective exploratory research, towards single sample processing and data analysis, including real-time test reporting for each study participant, with the respective assay design implications. Assessment of technology, in our case qPCR, does also play a role, and the question arises if one should use an IBD-grade instrumentation platform. And very importantly, in which countries and regions do we anticipate our clinical trial sites to be, and how does that translate into regulatory requirements and with respect to legal and data protection implications, etc. If using an example, one has to think about the different regulations in the United States versus Europe, for instance, which will all impact assay development. And in addition, all these considerations will influence vendor selection, who will develop and conduct the test. This is just a high-level overview of the development trajectory for the NRS clinical trial assay, which we have developed together with our vendor. And I would like to highlight the fact that a prospective sample collection of the required sample type and in the intended use population is of crucial importance for biomarker assay development. Thinking early about sample collection of potentially different sample types, keeping in mind harmonized sample collection and storage conditions, controlled aliquoting and freeze-thaw cycles, that's all absolutely crucial and will drive success of assay development and potential future bridging strategies. Assay design input and assay development according to user requirements is another important topic in the fit-for-purpose development of a CTA. And in our case, and for several reasons, we made the decision to switch from single qPCR assays to multiplex design. Once the assay has been logged, the analytical validation studies can be performed. And simultaneously, the clinical trial assay reporting software also needs to be developed. And only once all these activities have been completed, the clinical trial sample testing can start. As mentioned previously, it is important to assess the regulatory requirements for each specific trial. But the idea here really is to have a generic fit-for-purpose biomarker assay, which is far enough developed so that it could be used prospectively across a series of different trials. Biomarker assay development is a complex topic, and one always has to ask the question, how will the test results being used and what is the benefit of using that test? And most importantly, how could this biomarker inform drug development and ultimately contribute to improved patient care and treatment outcomes for patients presenting with certain clinical signs and symptoms? So let me briefly recap all the information with you. I've shown you an example of a gene expression signature which works best for predicting non-response to therapy, and which we refined and trained and have taken through several retrospective assessments through currently 3 different mode of action biologics. The data suggests that the NRS is a prognostic gene expression signature, and it uses baseline colon biopsy material of patients with ulcerative colitis to predict if they will respond to treatment as based on week 8 endoscopic mucosal healing data. We want to use it prospectively in clinical trials, and as such, we have assessed the requirement to develop of what I would call a fit-for-purpose biomarker test, or with other words, a clinical trial assay. Of course, as with everything in life, there's always room for improvement, and thinking about second-generation tests, we do have simultaneous research activities ongoing, for instance, to move to a less invasive sample type in the future, if feasible. We also want to expand biomarker-based response prediction and patient segmentation into other inflammatory bowel diseases beyond ulcerative colitis, really with the ultimate goal in mind to provide better treatment outcomes for patients with IBD. But in essence, it's a long and winding road to walk. And with my last slide, I would like to thank you for your attention. And though I cannot share all the data, I would like to invite you to check out more detailed information of how the signature was discovered in the listed publications. So thank you very much.